MHC polymorphism affects antigen recognition by T cells by influencing both peptide binding and the contacts between T cell receptor and MHC molecule. The products of individual MHC alleles can differ from one another by up to 20 amino acids, making each variant protein quite distinct. Most of the differences are localized to exposed surfaces of the outer domain of the molecule, and to the peptide binding groove in particular. The polymorphic residues that line the peptide binding groove determine the peptide binding properties of the different MHC molecules. We have seen that peptides bind to MHC class I molecules through specific anchor residues, and that the amino acid side chains of these residues anchor the peptide by binding in pockets that line the peptide binding groove. Polymorphism in MHC class I molecules affects which amino acids line these pockets and thus their binding specificity. In consequence, the anchor residues of peptides that bind to each allelic variant are different. The set of anchor residues that allows binding to a given MHC class I molecule is called a sequence motif. These sequence motifs make it possible to identify peptides within a protein that can potentially bind the appropriate MHC molecule, which may be very important in designing peptide vaccines. Different allelic variants of MHC class II molecules also bind different peptides, but the more open structure of the MHC class II peptide binding groove and the greater length of the peptides bound in it allow greater flexibility in peptide binding. It is therefore more difficult to predict which peptides will bind to MHC class II molecules. And that's MHC polymorphism affects antigen recognition by T cells by influencing both peptide binding and the contacts between T cell receptor and MHC molecule. Have a great day.